Hello students, today we are going to discuss about the Mendelism. A question arises in our mind is that why the Mendel was selected pea plant as an experimental material. So, at the time of the selection of pea plant as an experimental material, the Mendel had certain consideration in his mind. So these considerations are the number one, variation. So the plants which were selected for the experiment, it should have a number of detectable differences. For example, the pea plant which was selected by the Mendelism, it has the 7 different traits. If we consider the first trait, that is the plant height, it has the 2 different categories, tall or dwarf. If we consider the flower color, it may be the purple or it may be the white. That means these plants which were selected for the experiment, it should have the detectable differences in their character, and at a time the single detectable character should be considered. The second consideration was the reproduction. The reproduction is most important thing for the recombination of the character. And for the genetic recombination, the sexual reproduction is most important, and therefore the plants which should be selected for the experiment, it must be reproduced sexually. So the chosen organism should be sexually reproducing, because only then recombination of characters from both parents will take place. The third consideration was controlled mating. The controlled mating means the cross pollination of the plants or the parents, it should be taken place in the controlled condition. So the chosen organism should be able to mate in controlled and when planned condition, because by this process we can produce the pure line homozygous parents. The pure line homozygous parents means what? The pure line homozygous parents means there are 2 categories, for example, tall and dwarf. The tall, it must contain capital T, capital T. The dwarf, it must contain small t, small t. So one capital T, capital is the homozygous dominant parent, and the small t, it is the homozygous recessive parent. Generally, the recessive parent is always homozygous. The dominant, it may be the homozygous or heterozygous. But when we consider the pure line, then it must be the homozygous. That means it should contain both the alleles in a dominant condition. Capital T, capital T. So, in control condition, when the mating will take place, then one should maintain the careful records of the offsprings for many generations. For example, F1 generation, F2 generation, and so on. So, it is most important. The chosen organism should be able to mate in controlled and well planned condition. The next consideration was the short life cycle. The chosen organism should have very short life cycle. We know that the pea plant, it has a very short life cycle, only 3 to 4 months. So in 3 to 4 months, the experiment will get over and we will get the results of that experiment. The next consideration was the large number of offsprings. The organism which have been chosen should produce large number of offsprings after each successive mating. It will help to deduce the correct conclusion. If we observe the sweet pea plant, it is observed that The single plant, it produces the number of pods, and each pod it contain number of seeds. So the experimental plants it produces the large number of offsprings, and this large number of offsprings, when we cultivate further, it will give the more correct results for withdrawing of the conclusion. The next consideration was the convenience in handling. So, it is observed that the plant which is used for the experiment, it was the pea plant, and generally the organism which is used for the experiment in field or in laboratory, it must be convenient and inexpensive. For example, if we choose the elephant as experimental organism in the laboratory, it is quite difficult. Why? Because it has very long reproductive phase, very less number of progenies are produced, and it is not easy to do experiment with the elephant. But instead of that, the Drosophila, pea plant, tomato, rat, guinea pigs, these organisms. They have the short lifespan. They reproduce in abundant number, so we can use it very easily. So Mendel found that pea plant is the best material for hybridization experiment, and as well as the pea plant has the 7 contrasting characters. So Mendel studied 7 different traits. The number 1, stem height. Number 2, seed color. Number 3, seed shape. Number 4, pod color. Number 5, pod shape. Number 6, flower position. 7th character or 7th trait, it is 7a and 7b. 7a is a flower color and 7b is a seed color. So first trait, stem height, it has the 2 distinguishable character, tall and dwarf. Tall is the dominant character, dwarf is a recessive one. Seed color, it is yellow and green. Yellow is dominant, green is recessive. The third, seed shape. The seed shape, it may be round or wrinkled. Round is dominant, wrinkled is recessive. Pod color, it may be green or yellow. The green is dominant in case of pod and yellow is the recessive. While in case of seed color, the conditions are reversed. Seed color, yellow is dominant and green is recessive. Then pod shape. Pod shape, it may be inflated or constricted. Inflated means the smooth surface or plain surface, and constricted means it has the beaded appearance. The inflated is the dominant character and constricted is the recessive character. The next, flower position, it may be the axile or it may be the terminal. So axile is dominant and the terminal is recessive. Then flower color. Flower color, it may be the purple and white. The purple is dominant, white is recessive. 7th B, it is seed coat color. It may be the colored or white. So colored is dominant and white is recessive. Besides this, the structure of, if we observe the structure of the flower, it has the large petal at the back side, that is the vexillum, and we can open that flower very easily. So that structure, it is satisfactory for the hybridization. So when we easily open the flower, we can easily remove the anthers from the flower to avoid the self-pollination and to promote the cross-pollination. This process of hybridization, in which the removal of anther to avoid the self-pollination, is known as the emasculation. So the process of emasculation is very easy in the pea plant because it's flower shape. The pea plant is a seasonal plant. It having 3 to 4 month life cycle. It produces many seeds. So Mendel had chosen this plant material. The Mendel has performed the different experiments. It may be the monohybrid cross, dihybrid cross, trihybrid cross, etc. So first of all, the monohybrid cross. So what is meant by the monohybrid cross? The monohybrid cross means when a single trait is considered at a single time for study of hybridization is known as the monohybrid cross. For example, the Mendel is crossed two homologous pure line tall plant capital T with the recessive dwarf plant small t and all F1 progenies it had shown tall characters. So it is the monohybrid cross. On selfing of the F1 progeny, that means when tall plant is crossed with the dwarf plant, in F1 it produces all the tall characters. So all these tall characters which are produced in the plant or in F1 progeny, they are heterozygous. That means they contains the genotype capital T small t. On selfing of this F1 progeny, both characters tall and dwarf appeared in F2 generation in 3:1 ratio. The cross, the two parents, there are two parents tall and dwarf. Tall is male and dwarf is female. It may be the vice versa. The tall may be the female, dwarf may be the male. Okay. So it may be the reciprocal cross. So tall contains capital T and dwarf contains small t. The tall it produces two types of two gametes. It produces the gamete capital T. Dwarf produces the gamete small t. When these two plants they crossed, they produce the F1 progenies and all F1 progenies were tall plant, but that tall plants were the heterozygous. That means they contain the capital T small t. So to check that whether all these are the homozygous or heterozygous, the selfing is allowed. So on selfing, tall plant of F1 tall plant of F1 progenies they are crossed. They produce the two types of gamete capital T small t. When these gametes they put in the Punnett square, it is observed that the one progeny it has the homozygous tall capital T. The two progenies they are heterozygous capital T small t, and the last one dwarf, that is small t. So phenotypic ratio was 3:1 and genotypic ratio was 1:2:1. So one is homozygous tall, two are the heterozygous tall, and one is homozygous recessive. The Mendel has stated the different laws. So there are the three different laws. Out of that, the first we are going to discuss here the law of dominance. The law of dominance is stated that the dominant allele always expresses over the recessive allele. So the cross between two plants differing in a single pair of contrasting character is known as the monohybrid cross. Okay. For example, the tall character, dwarf character, the single character that is the plant height. When we crossed this tall and dwarf, it produces the results we already discussed it. So Mendel made a monohybrid cross between tall and dwarf plant Then only tall plants, they were appeared in first filial generation, that is F1 generation. But when F1 progenies were allowed to be self-fertilized, both tall and dwarf characters were appeared in F2 generation. This shows that in F1 hybrid, the character of tallness, it dominates to the character of the dwarfness. And so the character of dwarfness could not express itself in F1 generation, and so in F1 generation all the characters were tall. So the character which expressed in F1 generation is called as the dominant character. And that was a law of dominance. The second, it is not the law, but it is the experiment in complete dominance. Sometime in a heterozygous dominant allele, the dominant allele, it does not completely mask the phenotypic expression of the recessive allele. And the F1 progenies, they produce the intermediate character, because we know that the F1, it has the heterozygous condition, so it produces the intermediate conditions. For example, the red is crossed with the white, it produces the F1 plants, they are pink in color, so that is the intermediate color. So it is known as the incomplete dominance. Here the example, when the red flowered pea plant, it contains the R pure line plant, is crossed with the white flowered pea plant, the smaller. Then the F1 hybrid plant were found to have pink flowers. It shows that the gene for red color could not completely dominate the gene for white color. In such a case, F2 phenotypic ratio and genotypic ratio are the same. F2 phenotypic ratio, it is 1 red, 2 pink and 1 white. So F2 genotypic ratio, 1 R, 2 R smaller and 1 smaller. Here the cross, the parent red and white. Red, it has the R, white smaller, produce the gametes R, white produce smaller. Then the crossing between these two parents, it produce the heterozygous F1 generation, R smaller. But the characters which are produced by F1 plants, they show the intermediate character between red and white. That means they are not the red, not the white. They are intermediate in character. And then on selfing, between that F1 progenies, pink versus pink, it produces the gamete R smaller. When we put in the Punnett square, R smaller, then the first progeny R was red, R smaller, two were the pink and smaller, one was white. So the phenotypic and genotypic ratio was the same here, 1 red, 2 pink and 1 white. I think you got the points. Thank you very much.